Hello and welcome to another bonus video. Many major crop traits such as yield, quality, stress tolerance, and certain forms of disease resistance are controlled by many genes, and are also known as quantitative traits. The regions within the crop's genome that contain these genes are known as quantitative trait loci, or QTLs. Many markers have been found over the years that can be used as a means of determining if these genes are found in a genetic line. In our current era, single nucleotide polymorphisms, or SNPs, are the marker of choice. SNPs are genetic regions found primarily in the regions between genes. Specific regions are inherited in such a way that a series of linked genes are inherited alongside this region. This means that specific SNPs can be used as indicators to determine the location of a QTL. Genetic markers are usually made or validated via a series of full or half siblings in a perennial species or a real population in a sulfur species. SNP markers are currently mostly generated using a methodology called genotyping by sequencing, or GBS. In common plant genetics and breeding programs, this is outsourced to other groups. GBS genetic datasets have a broad spectrum of uses compared to prior methods. The process of creating such genetic datasets are complicated and will be explained in a future video. The common software packages used to analyze this material would be ICI mapping, and this software package will be explained right now. This is the data set, and this is ICI mapping, as you can see right here. This represents all the data in the practice program that represents fictional data. As you can see, this is all the data, and when we get this to start, the program analyzes both the practice phenotype data and the practice genotype data. As this is an example program that will be running through this process. And then as a result, we get the genetic map. This is a list of chromosomes being studied. And we can also show the map. Took the wrong turn. There we are. As you can see right here, these are the different genetic regions that are studied within this practice piece of data. This line right here represents the minimum amount of LOD score, which represents the statistical likelihood of this gene being a causal player within the trait of interest. This line, or hump, as you can see right here, indicates that these genetic regions around here are likely sources of a trait of interest. The farther up the line goes, the more genes of interest can be found. Well, the more likely the genes of interest can be found. As you can see right here, very high LOD score. So these three genetic regions listed here are very likely players in the trait of interest within this practice dataset. as are these genetic regions. This one a little bit less likely, another is a little bit more likely. The regions of interest that have lines or humps associated with being below this proverbial line indicate that this trait of interest is not likely connected with those genetic regions. And this basically is how this whole process operates. The software does the heavy lifting, and then we interpret the number by looking for traits of interest that are above the line. Well, specifically genetic regions of interest that are associated with a specific trait, and finding them above the line or below the line. The more above the line they are, the more likely they are to be associated with that trait of interest. This is a more comprehensive look at it as well. You can see it looking pretty good. The previous one had the additive effect, which means that the trait is known to be additive, which means if you combine with another trait of interest, then the result will be an improvement as well. Another genetic region of interest, I mean. But it's easier to see with just a lot score. And as you can see, these three regions right here are pretty significant players based on this genetic dataset. So are these three players. And anything below the line, probably not. Thank you for hearing me stumble through this little lecture. Back to the pre-record stuff. Thank you for bearing with me stumble through that little explanation. This is why I often use pre-recorded videos that allow me to edit a lot easier, but that's not here nor there. The rest of the video will be split across 3 sections. Section 2 will cover disease resistance, and we'll use flax and apples, covering Fusarium and fireblight respectively, in terms of their QTLs. The A-box stress tolerance section will cover peaches and wheat, specifically covering cold hardness and drought tolerance respectively. The last section will be dedicated to quality traits. The crops used as examples for that section will be lentils and cherries, covering zinc and flavor respectively. Within the realm of disease resistance in crops, there are 2 forms of disease resistance: quantitative and qualitative. Qualitative disease resistance is controlled by a single gene. This form usually creates absolute immunity to a specific set of a crop disease strain. This form comes with 2 major weaknesses. One weakness is that only a certain set of diseases have corresponding single gene resistances. For instance, there are many such genes for rust and none for Fusarium. Another weakness is that the immunity created by that gene only lasts as long as that specific set of strains predominate. Since pathogens are prone to mutation, novel mutant strains of the disease will appear and render the gene obsolete. This means that a new gene needs to be found to replace the old one. Quantitative disease resistance is controlled by multiple genes, is environmentally dependent, and does not create a full immunity, but is durable and can last pretty much forever. Breeding for the former is easy, but the latter is hard to breed for. QTLs make the process easier, as you do not need to use long periods of disease screening that usually take multiple years in order to find resistant strains. Instead, you can just use the genetic marker and you'll be done with it. For this episode, we'll be covering disease resistance QTLs found in flax and apple. In flax, the cultivar Bison is the most predominant source of Fusarium resistance in the Canadian flax cultivars. This cultivar was first bred for that specific purpose in NSU in the early 20s. Much of the early generations of Canadian flax cultivars were bred from Bison, and these would be the origin points of Fusarium resistance in much of the Canadian gene pool, particularly the CDC and AAFC breeding programs. CDC in this context represents the Crop Development Center, the University of Saskatchewan breeding program for wheat and oilseed crops. The AAFC is the government breeding program for different oilseeds and grain species. In the genetic study by Dr. Booker, Bison would be crossbred with a susceptible flax cultivar called Novelty to breed 703 rills. These were genetically tested and evaluated for Fusarium resistance. The Fusarium data would be combined with the genetic data using ICI mapping software. We went through the whole process of how to use this in the last section. The phenotypic data, which is to say how well they resist Fusarium, and the genetic data would be combined using that software package. Through this process, it was discovered that the QTL, known as LUS 1002589-1, was the source of the Fusarium resistance genes within the cultivar Bison. For apples, fire blight is a major bacterial disease and one of the few that can actually kill the tree. Over time, various researchers would discover different QTLs. One of the major QTLs is the genetic marker GE8019. This QTL is a genetic region first discovered in the cultivar known as Cox orange pippin. It is also found in many of its derivatives and several other unrelated cultivars. The next slides will show some of the cultivars of this QTL. Only those available in Canada will be listed. Another two QTLs of interest are the FL0 and FLO10 QTLs, which are found in Red Delicious and Jonathan respectively. Both regions have good combining ability with each other and have other resistance sources, and both play a role in improving fire blight resistance. From the perspective of abiotic stresses, there are various stresses that are not disease-related. Drought, salt, heavy metal, and cold all play a role in a plant's survivability as stress agents. In wheat, drought is a major player in a long list of abiotic stresses. An experiment will be done where two cultivars, C306 and HUW206, be crossed together to create 104 separate lines. The former is drought tolerant and the latter is susceptible. Through the analysis of these 6 lines, 6 candidate QTLs were discovered, and of these regions, the marker XGWM337 had the biggest impact on improved drought tolerance. In peaches, chilling injury causes a great deal of damage to the fruit. Different studies have been made over time, oftentimes using a population derived from the self-pollination or crossbreeding of a cultivar known as Venus, a cultivar that can generate very durable fruit, though other populations were also used. Researchers over time would find 10 QTLs that reduced mealiness, 4 QTLs that reduced fruit bleeding, and 2 QTLs associated with fruit browning. Quality traits. Even a cultivar of all the traits of interest regarding stress tolerance cannot survive without the traits that make a cultivar tasty and healthy to eat. As a result, many researchers have studied the QTLs of quality. In cherries, various forms of acid and sugar play a role of creating the flavor of a sweet cherry. Many studies would find such QTLs. One was an unnamed QTL but has an analog in other Prunus species, indicating that this region is likely found in a common ancestor group containing the cultivated species of Prunus. Dr. Gracia would analyze 5 populations of sweet cherry derived from the crosses and self-pollination events of different cherry cultivars. These were Lambert crossbred with Chrysabella, Vic crossbred with Chrysabella, Ambruns crossbred with Chrysabella, a Celphane of Chrysabella, and a Celphane of an experimental cultivar called BC-8. Through this analysis, several QTLs for different sugars were found, including glucose, sucrose, fructose, and sorbitol. Different QTLs associated with different acids were also found, such as one for malic acid, quinic acid, oxalic acid, and shikimic acid, all of which play a role in making sweet cherries taste great. The corresponding QTLs can be seen on screen right now. Dr. Kirsten Bett would also study many cultivars of lentil found worldwide to see which have the most iron and zinc, both of which are nutrients that many people are deficient in worldwide. Through an analysis of the genetic and phenotypic data, it was found that increased iron content is highly heritable in a species, but zinc is only moderately heritable. The QTLs LC C25 737P350 and LC C2 4316 P626 were found to be highly associated with an increase in iron content in the seeds. The QTL LCC 05435P444 showed the strongest association with zinc concentration, although the link was weaker than the QTLs related to iron concentration in the lentil seeds. We'll conclude with a bit of speculation. The cultivar Bison is possibly a derivative of the landrace known as Dutch Rigged Child. And if this is the case, many more QTLs or genes found in the resistance QTL in Bison could be discovered within this landrace. Likewise, in apples, Dr. Oldenburg, not to be confused with Duchess of Oldenburg, is a hybrid between Alexander and Ananas Renette, both carriers of the GE8019 marker, and both Gascoigne Scarlet, Peace Good Nonsuch, and Wolf River have Alexander as a parent, and they may be carriers of that marker as well. Cox Orange Pippin and Ripson Pippin both have Margell as a parent, but Margell is susceptible to fire blight, possibly indicating that the heir parents are the sources of this resistance. Rosemary Russet is a second pair to Cox Orange Pippin, and an experiment by Dr. Luby has found that it has the ability to pass on resistance to its progeny, indicating that Rosemary Russet may be the source of the fire blight resistance within the Cox Orange Pippin genome. Since Ripson Pippin also has a GE8019 marker, the second parent, Nonsuch Park, is plausibly the resistance source. But that's enough speculation, and that about covers everything. As always, the sources are listed in the description box below, and thank you for watching.